Oh, sorry. Should leave this here. All righty, everybody. Well, welcome to the Aquarium of the Pacific. My name is Callum Watson. I am the Youth and Family Volunteer Coordinator here at the Aquarium, and I am super excited to share with you our Teen Science Café event. We have a great audience here today, and then we also have some folks who are tuning in from home. I know it's been kind of rainy in this California weather, so watch from home, but it's good to have you here with us today. So we are going to get started with our first Teen Science Cafe program for the year, and we have only a few left of this series, the Antarctic Women in Science series. So if you are ever interested, come and join us. It's always a good time. We get to ask some really interesting questions that you might not ever get to ask an Antarctic scientist ever. So please come and join us for these events. We have a great time. Great, so before I start, there's nothing else. Who wants to start us off today for our Teen Science Cafe program? Is that going to be Jay? Okay, great. Come on up. Thank you so much. Let's get started.
Hi everyone, my name is Jana Lee, or may call me Jay, and I am a part of the Teen Science Cafe program. And for those who do not know what the program is, 1 second, can we get the next slide? Oh, is there a clicker? Oh, here it is. All right. Thank you. So we are a group of, so we have our presentation today on antifreeze ice fish that are found in Antarctica. So we are a group of teens who organize events for the teens local to SoCal, or those who are joining us online. And we have a speaker and we bring them in to talk more about their own research and conservation efforts, as well as more about our marine life and organisms that are living on our oceans or in our oceans. So recently we had multiple talks, part of our Antarctic Women Researchers, our series, and our first one was with Dr. Amy Moran on Antarctic pygmalionids, and Dr. Margaret Ansler with pelagic shrimp, Dr. Roxanne Beltran with Weddell seals, and Dr. Kristen O'Brien with Antarctic ice fish. So now I would like to pass it on to Kate, who will talk a little bit more about Antarctic conditions.
All right, so before we get started with the Antarctic ice anti-freeze fish, let's talk about their habitats. So, the Southern Ocean. There's a belt of water around the Southern Ocean, which is called the polar front. And it's also known as the convergence. And as the name suggests, it's where most of the ocean water converges or meets. And in this area, the warm water from the equator moves down to the Antarctic in the Southern Sea. And because warm water is less dense, it'll rise to the top. So, but as it gets closer to Antarctica, it'll cool down. And as hot water cools down, the density increases and sinks to the bottom. This loop creates currents and moves the Earth's water around. And going back to the Southern Sea, there is over 11 million square kilometers of sea ice, 1 to 2 meters thick, that surrounds the continent. And the temperatures in this region can range from about -2 to 10 degrees Celsius, but in the northern side, it can range from 7 to 12 degrees Celsius, which is a little hotter. And in the southern side, it can be about -1.9 degrees Celsius. And now I'll be passing it off to my friend Ditya.
Hi, my name is Ditya, and I'm going to be talking to you guys about Antarctic seasons and how, seasons vary, well, here versus Antarctica. So yeah, up on the slide, as you guys can see, there's a chart there, and I'm just going to ask you guys a couple questions about the chart, see what you guys can infer from it. So the first question is, what do you notice about the months? So, like towards the left, you can see the different months and how the seasons correlate, I guess, and so what do you guys notice? can raise your hand, or like I can call out. That's true. It's pretty cold there. Yeah, in the back. Yeah, that's like the biggest difference. Yep. Is there any other observations? Okay, cool. Next question. Yeah, we already kind of answered this one, but do they match the seasons in California? So, as we saw, the seasons and the months, they don't really match up from here versus Antarctica. So, yeah.
So a little bit more on that. So here is kind of an average chart. So it's not very specific where degrees would be the highest is 0 degrees Celsius, but it's kind of like a little snippet of what you might see there. It gets extremely cold. And what's important about these seasons is that in the summer, for example, February, here in California, we would have the summertime being in June, and this affects the cycle that they have in place, their life cycle, and the way they interact with climate change in general. So, can we have the next slide?
So, with all these extreme conditions, you might be wondering, how do fish even survive in Antarctica? Erica, do you know?
Well, there's a little something we can learn about first, and that will be freezing point. So freezing point is the temperature that a liquid becomes a solid. Think about water freezing and turning to ice. And on the next slide, we'll have some examples of freezing points. So first, we have fresh water. You guys might know that fresh water freezes at 0 degrees Celsius. But what's interesting is that salt water actually freezes at -2. And this is because when water wants to freeze, it forms ice crystals. And the problem is when you have a lot of salt in the water, it kind of gets in the way of those ice crystals forming. So you need a much lower temperature in order for that water to freeze. Some other examples are fish blood. Most fish blood freezes at -1 degree Celsius, and our blood actually freezes at -0.6. And we don't freeze at 0 like fresh water does because we have a lot of proteins and salts in our blood.
But something really interesting is that Antarctic fish have a freezing point of -2.5 degrees Celsius, which is a lot lower than normal fish or even humans. And that's because they're adapted to the Antarctic waters, which, as mentioned by Kate, is very cold at -1.9 degrees Celsius. And they do this by having antifreeze proteins in their blood that keeps them from crystallizing, and we'll talk about that more.
Thank you, Zoe and Erica. So proteins are very important for all of our bodies. As you can see on screen, there are several foods that are known for having a lot of protein. Do we recognize any? Is there a food that you know has a lot of protein even though it isn't on screen? What are we thinking? Yes? Vegetables like broccoli, that's true. They have a lot of protein. What else are we thinking? Yes? Eggs, that's a great source of protein. Are there any others that we recognize? All right, so there are a lot of foods that are known for having protein in them. But what exactly do proteins do for our bodies? Well, I think a more difficult question to answer would be, what don't proteins do for our bodies? Proteins are present in just about all of our body parts. They're present in our tissue, in our organs, in our muscles, in our bones, even in our hair. In every one of our bodies, we have thousands of different kinds of proteins, and every single protein performs a unique function that keeps us alive and healthy. Proteins called enzymes help to break down food and turn it into energy that our bodies can use. Proteins called hemoglobins help to supply our bodies with oxygen. There are many proteins that go into repairing tissue, to replacing it with fresh, healthy new tissue. And in the case of these Antarctic fish, they have a special kind of glycoprotein that behaves as an antifreeze for their bodies. Let's dive a little deeper into that word, glycoprotein. What is a glycoprotein? Well, the prefix glyco means to either be sugar or contain sugar. So quite simply, a glycoprotein is any protein that has sugar attached to it. There are many different kinds, but Antarctic fish have that special kind that behaves as an antifreeze and prevents them from freezing. This adaptation is absolutely essential to their survival in the Southern Ocean, because freezing is deadly for animals. When ice crystals form within the body tissue, it ruptures the cells and shreds the tissue apart. But the Antarctic fish have that special glycoprotein, which recognizes the ice crystals and prevents them from getting any bigger, saving the fish's life. Another important element to understanding glycoproteins is understanding how these fish maintain their body temperatures in the first place, which I'll invite Amina up to tell you more about.
Okay, as Vanessa mentioned earlier, the ice fish would be under the category of ectotherms, organisms that are dependent on their outside environment for keeping their body temperature at a stable level. The opposite of that would be an endotherm, which an endotherm organisms like penguins or like widow seals that have to rely on like their fur or their blubber to keep their internal heat levels at a temperature it will keep them alive. Can anybody tell me some endo or ectotherm organisms shown that are on the charts?
So we'll have like 2 minutes for you guys to discuss in groups a little bit so we can have some discussion flowing. I know you guys are a bit shy, but we want to see if you look at this chart and see if you can tell the difference between an ecto and an endotherm. So go.
Okay, on the chart, what do you guys think which is which?
Hi guys, so we're done with our discussion. So on the chart, which animals do you think might be an ectotherm? I want to see some hands going. Or I'm gonna pick out of the crowd. All right, the guy with the red, sitting next to the red sweater. What do you think might be an ectotherm? The colossal squid? Yeah, that would be an ectotherm. And that is because here, they're an organism that is dependent on their environment for body heat. So this squid might be dependent on its environment and its temperatures to maintain and change its body heat. Can we name an endotherm? Vanessa? The penguins, yes. And some of the penguins there are, well, actually all of them are endotherms. And that's because, like you mentioned, they're organisms that are capable of generating heat internally. Okay, I'm going to pass it on to Ditya.
Okay, now that we learned a little bit more about endotherms, exotherms, and what they are, I'm going to talk more about, we're going to come back to the antifreeze glycoprotein. So, as we mentioned previously, ectotherms are animals that need the environment for their body heat, and yeah, their body heat basically depends on the environment. And yeah, with the antifreeze glycoprotein, there are chains of proteins and carbohydrates that prevent the ice crystals from growing in their bloodstreams. That's basically what they do, the antifreeze part of it. And these proteins, they bind to the ice crystals that are growing in the bloodstream, and that inhibits them from becoming larger and larger and taking over. And yeah, you can kind of see these glycoproteins as the heroes of the story. So you have your blood and it's freezing up, and these glycoproteins really come in and they just save the fish in this case from freezing over. And yeah, when these animals freeze due to temperature, it's dropping, and like when they start freezing up and their blood pressures start lowering and these ice crystals start developing, this antifreeze glycoprotein, like I said earlier, is really just the hero, and they're the things that basically keep them alive.
So proteins, they don't respond well to heat at all. So what happens when a protein like comes in contact with a lot of heat? So the protein loses their original function. And this process where they lose their original function is called denaturing. So denaturing happens, as you can see in the image, is on the left is a protein, and when it's is with heat, it denatures and it returns to their primary structure. So I want to ask you guys a question. What things do you notice when you cook an egg? What happens to the egg? So, like, say you're going to boil an egg. Okay, Erica? It hardens up. Yeah, it hardens up. So basically, it becomes a liquid to a solid. And when you boil an egg and it turns into a solid, it can't turn back into that liquid, right? So that's just like denaturing. Once a protein denatures fully, it cannot be restored back to its primary function. And yeah, so next slide, please. So when proteins are, when the body is exposed to heat, there's something called a heat shock response. So a heat shock response is when basically the problem is that your body enters a state of stress, and this could happen from like really high abnormal temperatures. So the hero of our story is the heat shock response proteins, which are deployed once the body is in the state of distress. Then, in order to save the body, the heat shock response proteins hold on to the proteins to prevent them from denaturing. So, and once the distress is over and once everything can resume back to normal, the heat shock response proteins help the denaturing proteins refold into their proper structures again. But, however, once a protein fully denatures, it cannot go back. So these heat shock response proteins only help the ones that are partly denatured to come back into their original functions. So, and this relates to Antarctic fish because they aren't exposed to high heats. So they cannot produce a heat shock response. So I want to do a turn and talk, and I want to ask you, if Antarctic fish have a hard time creating heat shock response proteins, how will global warming affect them? Like, what happens during global warming?
All right, go.
Thank you. No one answered the egg. That's why I was saying I was motioning to say think. Yeah. Oh, like, yeah, because you have to really be pushy. Yeah, it's okay. Yeah, it's okay. That's not what it is. Okay, so now, does anyone have an answer to this? And if no one raises their hand, I will call on someone from the crowd. Ah, thank you. Yes, you in the orange jacket. Okay.
So he just said that they die, which is true.
correct. Which is correct. And why do they die?
Because the proteins that keep them from freezing over, those could they could damage a lot. So,
Yeah, all of the proteins denature, as I was talking before, and this causes the ultimate death of the organism. Okay, and we're going to do one more question. So, if a fish from outside Antarctica traveled to the Southern Ocean, what would happen to the fish?
Go.
Yeah. That's gotta be the perfect
All right, guys. So, does anyone have an answer? I know this is a kind of difficult question, but yes, yes, you. Are there? Yes, they said that the fish would also die because their bodies are not adapted to that cold water, and that's really true. So, what makes Antarctic fish so different than regular fish is when we talked about antifreeze glycoproteins, and that really helps them survive in this cold water. So, if a fish from outside the Atlantic Ocean, the Pacific Ocean, they come wandering in, they will freeze up. They will not survive that cold temperature. So that's why, it's really important to maintain that temperature for the fish that live there that rely on that cold environment. Right. Thank you for listening, and I'll pass it off to Callum.
Okay, very good job. That was a lot of information. So I know some of you in the crowd right now are like, where do I begin? Where do I start? Right? But we can do this together. While we're here, I just wanted to do a quick rapid fire fire away session here where we just recap everything we just learned, right? We did talk about some scenarios. What would happen, right, if some animals that live in the heat or in warmer temperatures that we are used to, right, they go down into Antarctica. They said they might die, they might freeze. What is the mechanism, right? Why would they freeze? What is it that we talked about that keeps them from, that wouldn't keep them from freezing? What is it? The glycoprotein, yes. Antifreeze, right? Does anybody here know antifreeze? You put that in your car sometimes. It's a little bit different, right, from a car and a fish, but keep that locked away in your mind as we get into our next presentation, right? Okay, another thing. Let's get into this one. So, it's getting hotter. It's getting hotter, and these animals down here in the ice, they don't need to worry about heat, right, because it's icy all the time. So, I don't need to evolve to have heat response proteins. So, what is it exactly that the heat response proteins do, right? Heat shock response proteins. What do they do? Yes. Yeah, denaturing. Yes, good. They keep the proteins from denaturing. What is it when proteins denature? What is that? What is that? I see your hand in the back. Yes. Yes, the proteins unravel and go into a different state, and they can't really function how they used to function. And we talked about a scenario, and you guys might eat this for breakfast every day if you're not vegan. What kind of food is that? Eggs, right? So when you crack the egg, it's all gross and slimy. You kind of have to like wipe it off, right? But after you do that and you start cooking it, you see it turn white. You see it turn a different color. The shape, the structure changes, right? That is protein denaturing. So all of those things, I want you to lock those away in your mind as we transition over to Dr. Cheng. If we can give her a big round of applause. And she will get a little bit more into Antarctic fish and her work.
Well, thanks, Caleb. And, this is very bright. Where's the best place to stand without that light? Okay, all right. It's an honor to be here, and it was really fun to seeing you guys. I think my undergrads can learn a few things from you, working so hard and learning so much, and so interactive. It's amazing. So essentially, you have given my talk, honestly, from your presentation. And I'm going to just tie all of those together and dig a little deeper into more sort of mechanistic things so that you have a big picture of how it all come together and worked in the Antarctic fishes, okay? So I come from the University of Illinois, Urbana-Champaign. If you don't know where that is, it is about 2 hours of driving from Chicago, south of Chicago. And is in the middle of cornfields and soybean fields. It's a very nice, peaceful town. The campus is quite pretty, especially in a nice fall day like this. Okay, so I've been there for about 40 years now. and I have worked with a lot of students, a lot of undergrads, and I'm here to talk to you about adaptation for cold in Antarctic fishes. I think I have to stand here so I can see better. And the antifreeze proteins, which you have talked about already, is really the key evolutionary innovation. And by that term, we mean this is a very, very important adaptation that has evolved that allow these fish to become very, very successful. All right. And so why do we want to study them? Well, actually, I want to show this picture first. This is a sampling of all the undergrads that have come through my lab over the course of many years. And you see a lot of girls, right? So girls can be scientists, right? So go for your dreams. And a couple of them, this guy here and this one here, actually went to the Antarctic with me. And then went on to do other things. She then did a master's with me, and then she's now in charge of the sequencing core facility at the Smithsonian Museum of Natural History. Right? And he has become a bioinformaticist working in data science, from learning how to deal with big data like DNA sequences and stuff like that. Okay. All right, and so it was very fun to work with these young people, and I can see all the enthusiasms, and the universities need kids like you guys. I shouldn't say kids, because I've been actually got into trouble by calling my undergrads kids because they think that I'm not treating them as an adult. But you are, you guys are young, right? Okay. So, have fish will travel, and I do go to the ends of the world to do my studies. The prior speakers have already told you where they go, and I go to the same place. And so there are these two main stations that we go to. So from Illinois, you fly, by commercial air, and Champaign-Urbana only has one airline that flies out of there, which is American. And so we fly American and all the way down to New Zealand, down here, this island country. And then from New Zealand, the U.S. National Science Foundation has a headquarters, and from there we fly by military flight. This is run by the National Air Guard. This is a C-17, and you get packed into that plane, and then you fly down to McMurdo, and you land on sea ice. Right, McMurdo is the biggest research station in Antarctica, in the US as well, actually across the world. The other place I go is where Dr. Christina O'Brien goes, and that is, and also Maggie Amsler, and that is the Palmer Station. And for this one, you also fly down commercial all the way to Chile, the southern town called Punta Arenas, and then you get on a ship. And then the ship sails down through the Strait Passage. Right? So that's between the tip of South America and the tip of the Antarctic Peninsula. And Palmer Station is right there. And it takes about 4 days to cross that Drake Pass, which can be extremely, extremely turbulent. My first cruise down there, we ran into 100-foot sea, and I was so sick. It's not even funny. It's like hell on earth. You cannot escape, right? What do? You jump ship, you're dead. All right, and so you have to be seaworthy if you want to do marine science on a ship. And I've eventually learned how to deal with that. Okay, and then I also go to the Arctic sometimes and catch fish along the northwest Greenland coast, as well as the north coast of Norway. And the reason is because not only Antarctic fish have antifreeze proteins, many fish species in the North Polar region face similar freezing conditions, and so they also evolve antifreeze proteins. Okay, and so I actually do go to both poles, and sometimes my kids call me bipolar. I guess that's right. All right. So the Palmer Station, you might have seen pictures from Dr. O'Brien already. It's a rather small station, so right by the sea. So I call it a seaside resort. It is really quite nice to go there, and it can house about 60 people, scientists and support staff together. And as you see, the station is surrounded by ocean water, and the whole Antarctic Peninsula coast is, of course, in the Southern Ocean. And so fishing there involves like going on the ship and setting traps and setting pots or trawling with nets. And this fellow here is Art de Vries. He's the discoverer of antifreeze protein in the Antarctic fish. And I joined his team, and he became my husband. And this was in 2017. He was already 74 years old. He doesn't look 74, did he? And so he's still going. You know, he's a really remarkable person I learned a lot from. All right, so fishing along West Antarctic Peninsula coast is open water, and these are the latitudes that we fish from. If you go to McMurdo, which is where I go most of the time, the environment is quite different. All right, so McMurdo Station is this big, sprawling station with many huts, many buildings. Right, it's kind of hard to see here, but anyways, there's a giant lab right here, yes, right here, and then these are dorms. And then in the height of the austral season, you know, summer season down there, this station can support 1,000 people. And so that's like rivals with a small U.S. town, right? Maybe even bigger. And can you imagine supporting 1,000 people? Everything has to be brought down there. It's a massive operation. All right, so the difference in environment between McMurdo and the Palmer Station, or West Antarctic Peninsula, is this. Here, you see ice. Under that ice is ocean. All right, so how do you catch fish through this ice? You do what the Minnesotans do. You drill ice holes. Okay, so and that's what we do, literally. And so this is at Cape Evans. This is like a bit away from the station. This is, well, a lot of, in the early explorers, especially from UK, uses McMurdo Station, or McMurdo Ross Island as the launching pad. And one of the very famous one, who suffered a ill fate, essentially died during his exploration, trying to get to South Pole, is Robert Falcon Scott. And they built huts when they are at McMurdo, and so Cape Evans, there's a hut there, and this whole place, this cape here, is named by Scott in honor of his second in honor of Edward Evans, who was the second in command during one of his expeditions. So, anyways, we use these gas-powered drill. You know, we drill maybe like a 4-inch hole. And then you fish, right? You just drop a line. You know, essentially, we actually just go to Walmart or Target to get these fishing poles, and you drop a line down that hole, and wait and jiggle and wait, and then you pull up fish. It's really cool. This is the only place on earth where you can catch fish so easily. All right, and so you catch small fish like that. You drill small holes, you catch small fish to get this very giant fish. There's one species that got to be really big. You have to use giant machinery. And so this is a giant reed drill that drills like a 42-inch hole. And then once it's punched through this giant hole through the sea ice, and that sea ice that is sitting on top of McMurdo Sound, which is ocean water, can be as thick in some years as 20 feet. All right, so it needs massive power to drill through that hole. And then you pull this hut over, and the hut has a hole on the floor, and it sits over this ice hole, and that's how you catch this giant fish. All right, and so you set down a line, and this is that giant species that we call the Antarctic toothfish. And I've used this species for many years because it's huge. It gives me a lot of material. I can purify antifreeze proteins from its blood and do a lot of biochemical studies. All right. So this is a short clip showing how this operation works. So that's the hole on the floor sitting on top of the ice hole. And this is a 1,000-pound stainless steel cable. And we fish about 1,500 meters down to catch these fish. And you need really strong assistance. And this fellow is actually a professor in physical chemistry who worked on the antifreeze protein structures and function with us. And it's very slimy. You have to we put them in a coffin like this with aerated seawater, and then and this is the size of it, okay? It's pretty huge. All right, and then we bring it, we keep some good specimens, bring them back in the coffin to the aquarium at the station and use them that way. All right, so here, so that's the show and tell. So here's some background on why we do the science. All right, and the Antarctic fish is extremely important because it's really the predominant group of fishes in the Southern Ocean. It has diversified to about 120, 130 species. And if you trawl along the coast, the catch is over 90% this group of fish. And so it's very, very important in terms of biomass. And so they became food for the penguins, for the seals. They became food for each other because they do each other, okay? And so they're a huge component, important component of the Southern Ocean food web. And so if you can imagine that when the Southern Ocean froze over, or it became very cold and freezing, and the fish had not evolved in a protection and survived, then the ocean will be emptied out of fishes, which would mean that the birds won't have any food, seals won't have any food, and the whole ecosystem would look very, very different today. All right? And so it's a very important group ecologically. And what happened that made them successful is that they have evolved a life-saving protection, which is this antifreeze glycoprotein that you've been talking about. And so studying how this happened would help scientists learn about the mechanisms of co-adaptation in ectotherm vertebrate animals, right? And there's a lot to be learned. And besides just the mechanisms, there's a lot of evolution involved. How do these antifreeze genes come about, which are novel genes with a very important new function. And then essentially they are just cool fishes, literally, it's my bias, sometimes they have the saying, that, the kid has the face that only the mother could love. And some of them actually are not that pretty, right? They're pretty drab, but they are cool from my point of view. All right, okay. And so a little bit more about the sort of environment that drove the evolution of these co-adaptations. This is Antarctica. If you see, this is there's this circulating Antarctic circumpolar current, and that polar front that you talked about is within this huge expanse of circulating oceanic current. This is the biggest oceanic current across the globe. And what's more important is that the water, this current is like a curtain that hangs all the way down to the bottom of the seafloor. Okay, and so basically it forms a barrier around the water around the Antarctic continent, and so we know that this is called Southern Ocean, and this barrier has two consequences. One, it separates this water here from the warmer water to its north. And so there is very little or no thermal exchange, and this is why over evolutionary time or geological time, that lack of thermal exchange led to glaciation over land and then eventually glaciation or in the seawater. And so this is a very harsh environment, and so because of this barrier, fish cannot get across either. All right, so that's the second consequence of this circumpolar current. And so the fish that live in here have to contend with that freezing, icy conditions. They cannot just say, oh, I can just leave and go to warmer places. They just can't, right? And so unless they can invent a solution, they will die out. And if they have not invented a solution, as I've said, then this whole ocean will be emptied out of fishes. And in fact, that's what happened about 40 million years ago. Before then, there's actually very many different kinds of species from the fossil record. You have sharks, you have regular fishes. But then now, today, if a survey of the species diversity of fishes in the Southern Ocean show that there's only these neurothenioi fishes that is the predominant group and make up like 90% of the biomass. All right, all the other fish, almost all of the other fishes have died out. You do not find sharks in the Southern Ocean. So there's no, sort of top predator. A lot of fishes that you see in the north are not represented in the Southern Ocean. Right? And so obviously, these guys have become extremely, extremely successful for two reasons, and the reasons are twofold. One is that they have evolved this protection, which is the antifreeze protein. And the other reason is because all the other fishes have died out. All the other fishes that don't have or haven't evolved the protection actually died out, and so there's much, much less competition. Right? And so these fish can radiate out and colonize all the water niches, column niches. Okay, all right, and so if you can't go, it must evolve a protection or freeze and die, and this is a very, very, very strong challenge, a very strong what we call natural selective force. All right, if it dies out, then there is no evolution, and so the lineage disappears. Right, that's what is called extinction, and so I said that environment is extremely harsh for these ectotherm fish, vertebrate fish, and this is so you ask how harsh. So if you jump down into through an ice hole into the water of McMurdo Sound, and you shine a light into the water column, you see that there's a lot of ice crystals around, right? And the water temperature at McMurdo Sound, which is sort of the southern limit of marine life, is minus 1.9 degrees Celsius, or minus 2, let's round it off. And that is the freezing point of seawater, which you have talked about. This is when liquid water freezes into a block of ice, right? All right, and so this is the environment that the Antarctic fish live in. All right, and the danger in this environment, which they cannot escape, is this. Right? We know that fresh water freezes at 0. All right? So this is freezing point. Fresh water has a freezing point of 0 degrees. Our blood, which you already told us, would freeze at about minus 0.6, and this has to do with the amount of dissolved ions and molecules in the blood. Okay? And in fact, our blood, the plasma that carries our blood cell, is mostly sodium chloride. Okay? And we have enough to depress the freezing point to minus 0.6. And so that freezing point depression is proportional to that concentration of solutes in the plasma. All right, so marine water, open ocean water, has this amount of salt, and it has a freezing point of minus 1.9. And the Antarctic fish, right, so it has about this much of dissolved ions and molecules, and so the expected freezing point would be minus 1 degree Celsius. All right, and so what you see here is you have almost a 1 degree difference between what you would expect the Nolothenoi fish to freeze and the environment out here. And so how does it prevent freezing? It actually, the biggest problem is not just the temperature, it's also the presence of ice crystals around. And this difference of 1 degree is a very, very strong driving force for ice nucleation. These fish swim around in all those ice crystals that you saw in the last slide. And once it comes into contact, there's no way to escape it, no ways to avoid it. The ice would immediately nucleate the fish, and it will freeze solid into ice very rapidly. And in the next video, this kind of demonstrates how powerful and how fast that freezing process would take place. All right, so some young kids would be a bit disturbed by what I'm going to show you. I hope you are old enough not to be disturbed. All right, and we'll play this video. This is actually an excerpt from a HHMI, which is Howard Hughes Medical Institute, the educational department who did a short film about antifreeze proteins. So let's see if I could play this. This is a younger me and a dorky professor from a number of years ago. Can you click? You have to click that triangle.
Give us 1 moment.
Okay. This sometimes happens when I teach at the university. And it was like, oh my God, I gotta get this to work. Time is running out.
Tiny ice.
Roll back, roll back.
Crystals are growing into larger crystals until the fish has frozen, while its tank mate is unaffected.
That was the tail end of it. Can we start over?
And fish cannot live generally
Here we
in
go.
that sort of environment because the freezing point of the
So this
blood
is the
is
aquarium
higher
at
than the
McMurdo
freezing point of
Station.
seawater. Okay, because they have less salts in the body fluids than in the seawater.
Yet fish thrive in these waters. Ice fish and other species, all members of a group called the notothenianoids. To show just how different notothenianoids are from other fish, husband and wife biologist Art DeVries and Christina Cheng dunk one in water as cold and icy as the ocean outside their makeshift lab on Antarctica's McMurdo Sound.
So as you can see, this fish is doing fine at this freezing temperature in the midst of ice crystals.
Then
This is
DeVries
one of your
adds an
questions.
ordinary temperate waterfish, a New Zealand cod, to the same tank.
You can see it's not very happy. It's beginning to convulse.
Quickly, inside the cod, tiny ice crystals are growing into larger crystals until the fish has frozen, while its tank mate is unaffected.
Did you guys count the time? How many
Why
seconds
this
before you
happens
died?
baffled scientists until the 1960s when Art DeVries found antifreeze proteins in the blood.
Okay, this is what we call a powerful, extremely selective force that drove the fish to invent a solution, right? Otherwise, it would be just gone like the New Zealand cod. Okay, and so the solution is this. Antifreeze evolved, and it depresses the freezing point from what you would expect at -1 all the way down to -2.4, -2.7, depending on the species. Okay, and so now you have this flexibility, or this clearance of about 0.5 degree to 0.8 degrees. Seem very small, but ice will not grow until the body temperature of the fish is depressed, if it ever will be, down to -2.4 or -2.5, 2.7. And so these kinds of temperatures never happen in the ocean. And that's because water it has what we call very high heat capacity. It takes a lot of energy to be removed for it to drop its temperature. And so when seawater reaches its freezing point, it usually just stays at about its freezing point. All right? And so the fish would never actually encounter temperatures any lower than -1.9. All right? And so this is why, with this antifreeze protein, it can swim around in this huge swarm of ice crystals and would not have any problem, would not freeze solid like the New Zealand cod does. Okay, and so this is a marvelous evolutionary innovation. And so you ask the question of like, how do antifreeze work? Right? So you have seen the structure in one of your slides. It has actually a very simple sequence, and it's essentially these 3 amino acids called alanine, alanine, threonine that repeats over and over and over. Okay, and it comes in a slew of sizes depending on number of these repeats. Okay. And the key point here is that every third residue, which is the threonine, is attached to a sugar. Right? And sugar is water-loving, it's hydrophilic because it has a lot of hydroxyl bonds, all right? And that can hydrogen bond with the water molecules in the ice crystal lattice. All right? And so this is that hydrogen bonding is what we think how the antifreeze glycoprotein docks down on the surface of ice. And when it does that, then it prevents other water molecules from joining the ice crystal. And so the ice crystal will not grow any bigger, right? Will not expand. And so in the ocean, this depicts the McMurdo Sound. You have fish swimming around. You have sea ice on top that that where we drill holes through to catch fish. And then there's water. There's a lot of ice crystals in the water column. They get into the fish because fish drink seawater. Okay, and they also eat food that would be studded with ice crystals on the surface. Right, and so the internalized ice crystal is recognized by these antifreeze proteins represented by these green blocks here. A perfect ice crystal is hexagonal, right? So six-sided. And so the binding of these antifreeze molecules then prevent water molecules from joining the ice crystal, and then the ice crystal will not expand, and then the blood will remain fluid throughout its life. And so this is how, for example, this Antarctic ball notothyn, one of the Notothenia fish, lives happily in the platelet layer of the full of ice in the McMurdo Sound. So that is that small fish that is in the same tank where the New Zealand cod froze and died. Can we play this? And you can see sort of how icy this is. This is actually a dive rope that hangs down for divers to find the hole to come back up. And this is giant, massive amounts of platelet ice, like sheets of ice, right under the surface fast ice. And you see here, this is one fish with a pair of eyes, and there's another pair of eyes inside. And it would be nice if we have the audio, but and they actually get into these ice crevices to escape from, to avoid being predated by larger fishes or seals or birds. All right. Okay. And so that's a very kind of a nice story. I can't advance until it's done, I guess. Right. Look at how icy it is. Okay, so you guys all follow me so far? Any questions? All right. Okay, so these Antarctic fishes have become experts of living in extreme cold. And now we switch gear, as you have also, the Antarctica and its water around it are warming, right, under global climate change. And that is bad news for these cold-specialized Antarctic fishes. And why is it bad news? Because of that heat shock system, right? Or the lack of. All right, so this is data that spans 50 years from 57 to 2006. And you see Antarctica is really warming up. The redder it is, the faster the rate. All right, and so Antarctica, the continent, is sort of like divided into the West Antarctic and the East Antarctic by this series of mountains called Transantarctic Mountains. All right, West Antarctica is warming up very, very quickly. And so is the ocean that is along this Antarctic peninsula. The East Antarctic ice sheet is also warming, but at a lower rate. All right, but but eventually it might catch up. All right, and so the West Antarctic ice sheet is this giant, massive ice sheet. And so if it warms up, what happens is that there's warm water welling up from the bottom and melting out the glacier that is hanging into the ocean surface. And until now, especially there's a giant glacier here called the Thwaite Glacier that is sort of held in place by underwater ridges. But since it's melting from the underside, it basically is now floating above that underwater ridge. And so eventually that entire ice sheet might just slide into the ocean. And when that happens, you should not try to buy any real estate in New York or Manhattan or any coastline, because you're going to have global sea level rise of tens of feet. Right? So Florida might just disappear. All right, and so or Long Beach, for that matter. Okay, and so that's a global sort of calamity when that happens. In terms of the fish, this is the problem. We know, as you have already told us, heat causes damage to cells and cellular molecules. which can eventually could lead to tissue death and the death of the species. So how do organisms protect against episodes of elevated body temperatures? So throughout the history of evolution of life, just about all organisms have evolved the system of molecules that are called molecular chaperones, also called heat shock proteins because they were first discovered to be present under heat stress of Drosophila many years ago. Okay? And we have these molecular chaperones in all our cells, and they don't just respond to heat. They are there because we continue to make new proteins, and new proteins have to fold properly. And so these molecular chaperones in your cells essentially help new proteins to fold, and then they can become the new set of proteins to perform function. All right, and the heat shock response is that it's like this, okay? It's sort of depiction of what happens. The proteins are there. What is missing in the Antarctic fish is the heat shock response, all right? And so these are regular normal proteins that have the good structure for doing its function. So you can think of the protein like lock and key. It has a very specific structure that can bind its target molecule, right, in a very sort of nicely fit way, and then carry on, and you can open the door, which is the function. Right? And when heat is applied or any other stress, it could be other sort of heavy metal poisoning and stuff like that. Those are also stress that can denature protein. Then you have the protein start to unfold. All right? And so normally in your cells, most of the heat shock protein, and I will use just HSP70, heat shock protein 70. 70 refers to the size of that protein. 70 means 7,000 molecular weight. Okay, and this is the key heat shock protein that is responsible for the so-called inducible heat shock response. All right, so normally in your cells, these proteins are are sort of associated with this other molecule called HSF. HSF is heat shock factor. And the heat shock factor is what we call a transcriptional factor that when it binds to its target gene would activate the expression of that gene. Right? And so there's low levels of heat shock proteins around, and many of them are associated with this heat shock factor. And so when there's a stress and some protein starts to unfold, and the increase in the concentration of the unfolding protein would be snatched up would promote the dissociation of HSP from the heat shock factor, right? And the HSP70 then ties up these, hang on to this unfolding protein. And so when it dissociates, the HSF would trimerize. They form a trimer, and this is the active form of that transcription factor. And when you trimerize, then it translocates into the nucleus. So your nucleus is where DNA is, right? Where all your genes are. And so gene expression happens initially in the nucleus. And so this active form of HSF gets into the cell. It binds to a recognition site in its target gene. So I'm showing a HSP70 here. And it activates its transcription. That's the first step in gene expression. And this is done in a massive way, so you have a massive increase of HSP being made, gets dumped out into the cytoplasm, and tie up all these unfolding protein that is accumulating in a fast rate under heat stress. Right? And so when that heat stress is passed, then these unfolding proteins, which are prevented from completely unfolded by HSP's help, then can start refolding also with the help by the HSP, and then going back into the functioning protein. And for those that are beyond, that are way too damaged, then there's a system of channeling them into degradation, so the cells can get rid of these dead protein. Okay, and so this system is extremely important in helping animals, cells, and therefore the animals to get through episodes of elevated body temperatures. Now, unfortunately, we know already that the Antarctic female fishes have lost this protective heat shock response. Right? And the idea or the hypothesis is that because they have not seen thermal variations through millions of years, there's no need to maintain this expensive system, this inducible heat shock response system, because they don't need it, right? And so if you don't use it, you don't need it, you lose it. And this is what we think has happened. All right, and so that's the problem. How do we know that it has lost the heat shock system? We have done tests. And so you have heard from Dr. O'Brien about this critical thermal maximum, and essentially it's heating up this fish to see how much heat stress they can tolerate. You warm them up until they reach a point that they lose balance, and then you immediately take them out and put them back into their normal water and let them recover. They don't die from it, so we're not sad about it. And so by doing these kinds of studies, we found that the Antarctic species have much lower critical thermal maximum values, right? About 10 to 12 degrees Celsius, much lower than its South American relative. Right? This one lives in temperate water, and it can tolerate up to 30 degrees Celsius without dying, while these guys can only tolerate up to a third of that. So we know that they are very specialized to cold. All right, and so after we've done these experiments, then we quantify the gene expression level of these heat shock proteins. And the test then will show if it has a lot of expression, then it has the inducible heat shock system. If it doesn't have hardly any HSP gene expression, then we know that it's either muted or silenced, right? Okay. And so this is what we do. This is kind of like a primer for you. This is a sort of regular gene structure for typical eukaryote genes. Right? So anything above bacteria. Right? And so we know that these are exons. Those are the bits of the DNA that code for bits of the protein. Right? And so the first step of gene expression is that there is transcription of this gene into an mRNA or messenger RNA, okay, and then it's essentially read off from the entire gene, and then it splice out the so-called introns, which are the non-coding part of the gene. All right, so now you have this complete gene, all right, with a complete coding sequence, and this is the mature mRNA. And then, of course, the mature mRNA go through translation to produce a protein that gives you the function. And so an easy way to test for gene expression is actually getting this part. We can isolate RNA from tissues, from all these CT-max treated fishes, as well as from the control that it has not been heat treated. And then we can sequence the RNA, the mRNA pool, through this new technology, massive parallel sequencing, and then you get huge amounts of data, and then you start doing, sifting through that data, quantify the gene, the level of expression of these heat shock protein genes, right? And so here are some results. So we compared the South American related relative that has a very robust. We know that it has a very robust heat shock response to an ice fish. Right, and to this ball notothon that is from McMurdo Sound. All right, so this number here is the total number of genes that have been induced by heat to express at very high levels compared to the unheated controls. All right, make sense? All right, and as you see here, there's a huge number of genes that are induced by heat to express at high levels in the temperate relative. In the ball notothon, there's only 19 genes. That's like pretty pathetically low, right? And interestingly, though, the ice fish has a lot of genes that were induced to express at a high level. So the question then is, like, of all these genes, how many of them are actually heat shock proteins, which is the genes of interest, right? And so you do more quantification. Oops, I think something got lost. Sorry. There's a whole panel of figures here that is not showing up. All right, and So just imagine there is a lot of HSP70s and HSP90s that are expressed by the South American relative among these 1,481 highly expressed genes, as well as a whole slew of other heat shock protein genes. And there's nothing to compare because from the Antarctic notothon, bald notothon, because there's only 19 and none of them is heat shock proteins. A few of the heat shock proteins are also expressed in ice fish, but the increase in expression level is very, very small. All right? And so this is how we know that the genes, the heat shock protein genes, are not expressed in the Antarctic fishes under heat stress. All right? And so these are the data that proves that the Antarctic notothenioids have actually lost their the heat shock response, the inducible heat shock response. So you would ask then, what are all these genes if none of them, if few of them are heat shock protein genes? They actually cover genes that are involved in inflammatory response, which are not involved in helping unfolded protein to refold, okay? All right, so given that, what kind of fate will Antarctic notothenioids fish under climate change? It really kind of depends on us, right? If we can slow down climate change, then there may be time for them to evolve to the changing environment, right? We have to slow it down a lot, right? So any animals can adapt to a changing environment because that's the natural selection process, right? And given enough time. And if there's not enough time and the change is too fast, too drastic, then the animal would not have the ability to keep pace, and then they would die out. All right, and so another encouraging sort of sign is that some of the species, several of these Antarctic species have ancestors that actually have escaped the circumpolar current. Right? And so you have one that is living in New Zealand. You have several that lives in South America, including an ice fish. So ice fish is particularly vulnerable. Why? Because none of them have heat shock protein, but the ice fish is particularly vulnerable to warming temperatures because it has no. What doesn't it have versus the other red-blooded fish? No hemoglobin. So they don't have that active oxygen binding and transport system. Yeah, right. So they're particularly vulnerable. But so even an ice fish, one of the ice fish, was able to establish in southern tip of South America. So there's hope, right? But these things happen over very long periods of time. You know, when there is sort of gradual changes in the DNA and in the genes that allow them to readapt to warm environments, right? And so the climate change then has to be slowed down drastically for that to be possible. So I'm coming to the end of this. So what have we learned today? We learned about adaptation, right? And so the Antarctic notothenioid fish have evolved AFGP. It avoided freezing and evolved to become experts at living in constant cold. And because of the constant cold, there's a trade-off. It lost its ability to mount the heat shock response. And so the fate is dubious, you know, because the lack of this protective mechanism, their survival would be jeopardized under rapid climate change. And so can humans slow down climate change to give them a chance to readapt? Otherwise, the Antarctic Ocean will be out of fish at some point. And then the penguins will be gone. And then the seals will be gone. That would be really bad, right? And so that would be extremely, extremely drastic loss of biodiversity, which we don't really want to see happen. So I want to thank all my grad students who have participated in postdocs on this research, and again, Art de Vries, who is my lifelong partner in personal life and scientific life. And I thank my son who did these beautiful images of these fishes for me. thank you for listening, and I'm happy to answer any questions if you have.
Yes, thank you again. Before we get into our question and answer session, I just wanted to wave goodbye to everyone who is watching on our live stream. Thank you for joining us. Of course, you're always welcome to join us again in real life, in real time. Come and join us. But you are always welcome to stay at home. If you're watching far away, that's okay. I know some of you are, even all the way up in Alaska. We see you. So yes, thank you for joining us. And we are just going to have a quick question and answer session. Okay, now that we are going to continue, great. Does anyone have any questions at all for Dr. Cheng? I do want to say this as we continue going on. As you go into college or a different program or wherever you want to go, right, if you want to study marine biology, you're going to learn a lot of different pieces of this before you get to this, right? This is a big compilation of many different subjects together, right? We talked about how different animal species kind of interact with each other, humans, ice fish, right? Rising temperatures, how that'll impact an animal, right? That's kind of biology, that's even ecology kind of, right? Then we even saw a little bit of exons and introns and gene expression. What even is that when our bodies make things, right? Who tells our body to make cells, things, right? That's genetics. So then you're going to have to learn about genetics. And then what you're going to end up doing is go on to put all these things together, maybe marine ecology, some other classes. You put all these things together, and then you start thinking of different questions. You say, well, I wonder what keeps these fish from freezing, right? And then you take all that knowledge together and you do a research. So this might seem like a lot. It was very interesting, might seem like a lot, but by the time you get into college, you're going to start learning these pieces, and it's going to be you doing this exact work, and you're going to sit back to yourself and say, oh, this wasn't as bad as I thought it was. So just look forward to it. Stick with it. You'll find it to be very rewarding, okay? So really great stuff. Any questions before we go into our activity for today? This was pretty thorough, and I think our presentation also kind of
questions about college.
Oh yeah, college experience, anything like that. Now's a good time to ask those questions.
Yes?
Do you have a favorite species that you enjoy researching?
Yes, but I haven't done a lot of research on them. I mean, they all have antifreeze, right? So I can use any species. There is one species that walks. Yes, I should have another video. It uses its pelvic fin. It walks very slowly. It's really, really cool. That's the coolest fish, yeah. And so you ask, when you see something like that, what is your immediate question? What would be your immediate question?
Are they related to epaulet sharks?
They what?
Are they related to epaulet sharks?
They are not. Oh, but you got a very good point. Yes. No, Antarctic fish is teleost fish, bony fishes. The sharks are very, very distantly related to this group. And so it's like, why do they need to, what is the force, evolutionary force or selective force that drove them to start walking? Yeah, and then how do they manage to do that? What are the genetic underpinnings of all this stuff? And so these are the curiosity-driven kind of thing that one gets sucked in.
On it yet?
Pardon?
Is there like no research on why they walk?
Nope. Well, I have done one study in collaboration with a functional morphologist, and trying to figure out whether its pelvic fins are any different from the other relatives. We haven't published that yet, but yeah, it doesn't seem to be that different, and so. And the thing is, like, we've discovered that it walks because when we found out that it has gobbled up another fish and the tail was still sticking out, I mean, it sucked in another fish. That's how they eat each other. And because the fish it eats is almost as the same size as itself, it takes time for it to, go in, go down several days. And when its belly is so full, that's when it starts walking. And when it has fully digested that fish, then it starts swimming again. So that's the one observation. Is that the reason? We don't know.
See, and that's where you all can come in and go down to Antarctica and do the research on that one.
Yeah. I think you have a question, right? Or no?
No, I was going to say that was an Antarctic one.
Yes.
I saw one, I don't remember where, there's another warmer climate one that does that.
Yeah, well, there are quite a few walking fishes. This is the only one that uses its pelvics. The others use like modified pectoral fins, like the gurnard. The fins become little fingers, very long, skinny things, and they crawl like that. Yeah. Yeah.
That would be so interesting to figure out.
Yeah. Yes.
Do you have an estimate on how long it would take for these fishes to gain their heat shock defense?
We don't know. So we're studying another species that has escaped to South America, and it seems like it has retained or has some heat shock response. So I don't know whether it's a reactivation of that inducibility or it reinvented. My guess is that it's a reactivation. And so as long as that system has not completely degenerated over time, then maybe it can be gotten back. The difference with the ice fishes is that in contrast, the ice fishes have completely lost the genes for hemoglobin. And so once you have lost those genes, then you just cannot bring it back. Inducibility has to do with those target sequence that the heat shock factor binds to, right? And so if that's a very small sequence that can go through mutation and revert back to functional size. And so it's possible, given time. So time is the main thing.
Good questions, everyone. Are there any more questions before we continue on? It's very interesting stuff.
Okay, I started going in 1984. Obviously, you weren't born. Maybe your parents weren't even born. So that was my first year. So I've been going for 40-some years. Usually about 2 months, 3 months. In the earlier days, it's about 3 months. And now they restrict your time down there because it's very expensive to support research. So you have, 2 months or so. Yeah. Question? No. Yeah, it's a very unreal world. It's almost surreal to be down there. You're sort of like, well, in the early days, in the 80s, before the time of the internet, snail mail is the only way. And if you can, if a letter gets down to McMurdo or back in 2 weeks, that is super fast. And so it's challenging that way. It's challenging for families. And so, that was, well, shortly after it started, we have 2 kids. And my husband is gone like 3 months out of the year, sometimes more than once a year. And then I'm home with these 3 kids, or 2 kids. And so it takes a lot to stay together. But then I go too, and he's stuck at home for a month. So he works. Oh, it's the most unromantic way you can imagine. So we are in the same department. I started as a graduate student. He started as an assistant professor. His lab is at one end of the basement. My lab that I'm doing my PhD is the other end of the basement. And so we kind of met in the middle in the basement. And so I've never left the basement since I got my PhD and started working with him, and then starting my own lab also in the basement. I never saw the light of day for 40 years. But time passes, and that's the basement has a direct route to Antarctica, so that's not so bad, right? Yeah, things sometimes happen in a weird way, or life happens in a weird way.
Friends from the back want to join us up front.
Yeah, that's good. Come on.
Yeah, come all the way up with us. Join us up here at the front. Okay,
That includes the pink hoodie.
while we do this. Okay, so thank you so much for answering all these questions. We're going to keep on working on this. In the meantime, I want you to turn to someone that you don't know, and if that's okay if you know them, but turn to someone you don't know, and I want you to think of three. Okay, see friend right here in the back. Come and join us up at the front. I know. Okay, I want you to talk about three big takeaways. If there are big takeaways that you could possibly take away from this whole thing, what would those three things be? Okay, and then I'm going to come back to you, and we're going to share out all together. Okay, so this is your time to network and find someone that you might not normally talk to, and just talk about three things that really stood out to you, okay? Ready? Go on ahead. I think Christina changed it.